Welcome to Careers in Discovery, your window into the world of leaders in pharma and biotech. Brought to you by Singular Talent, making hiring better for organizations involved in drug discovery and R&D. Clare Jones is the Chief Scientific Officer of Talisman Therapeutics, a biotech company committed to revolutionising the discovery of disease-modifying treatments for neurodegenerative diseases using stem cell technologies. Clare talked with us about her career in biotech, taking jobs where you both bring something and learn something, and the importance of clear, ongoing communication. This week on Careers in Discovery, I'm delighted to be joined by Clare Jones of Talisman Therapeutics. Clare, welcome to the show.
Thanks very much. It's great to be here.
Great to have you. So Clare, I always like to talk a little bit at the beginning about what you're up to now. And at Talisman, you're doing some very, very interesting work in neurodegeneration. Tell us a bit about that. Tell us what you're up to.
Yep. So Talisman's a small company based in Cambridge at the Babraham campus in the UK. We span out of Rick Livesey's lab at the University of Cambridge in 2013. And we're really focused on using stem cell models of neurological disease to help companies explore their questions relating to drug discovery. So we particularly like working with patient-derived iPS that we can turn into neurons, microglia, and astrocytes and different cell types that are involved in neurological disease. Because if you look at patient-derived cells, particularly those from patients with genetic forms of disease, they actually show cellular dysfunction, even at that early stage, that is also seen all the way through to post-mortem brain. So they're a really cool system to allow us to study in human cells the really early things that are going wrong in cells. And obviously our hope is that that can provide insights into the pathological processes that then ultimately lead to Alzheimer's, Parkinson's, Huntington's, all of these neurodegenerative diseases. And we can use these model systems to test new medicines potentially, or look at what's going on mechanistically.
Yeah, I see. And so hopefully start to diagnose some of these early, catch them before they cause real damage, which is part of the problem, right? Is they get caught very late.
Yeah, so it's more testing new medicines and looking at new targets. So it's new kind of ways of tackling the disease at an early stage. But yeah, absolutely, it's trying to hit the kind of early cellular dysfunction.
And am I right? This may be out of date, so correct me if I'm wrong on this. But am I right in thinking there's a particular focus on dementia?
So that's where it all started, yeah. Okay. So we started with the cortical neuronal models that were developed in the academic lab. We're now also looking at Parkinson's and a whole range of other diseases. So there's a lot of interest in lysosomal storage diseases. Kind of the rarer diseases are attracting a lot of attention as well, and looking at the overlapping biology and kind of where are there parallels between the different neurological diseases, and where do you need really specific therapies. So yeah, it started with dementia, and now we're kind of expanding out.
Yes, I see. And I suppose I always find neurodegeneration and therapies for it fascinating, because it's clearly an area of need and an area of quite unmet need in many cases, but it's just so complicated that it's hard to treat these conditions, right? So is there sort of, do you see the field making progress? Is there been, is that, clearly you guys feel you are, but in general, are we starting to break down some of the complexities around these diseases?
Yeah, I mean, you're absolutely right. It's really complicated. So that's a good starting point, but we like a challenge.
Yes.
I think there's lots of really interesting data coming out now, and there's an increased understanding, partly coming from these stem cell models that we and many other groups are using, that allow us to really explore what's going on in the genetic forms of disease. Now, obviously, there's also sporadic forms of disease, but I think that combined with the clinical trials that are ongoing, so obviously there's very high profile anti-amyloid antibodies that have been going through, with varying levels of success. There's also tau antibodies coming through, and I think with the focus on the clinical studies as well, actually, that even the failures are also informing, what are the approaches that have worked?
Yes.
Where are there kind of glimmers of things which do look like they're having an impact? There's lots more to do, but I think there's some really interesting work going on at the moment. So I'm optimistic, and I think we and others are really trying to develop models that will help support the next generations of therapies that are coming through.
Yeah, absolutely. And it's a good point. I suppose, although the failures can be disappointing, they're also instructive, right? And sometimes more so than successes, I guess.
Yeah, I mean, it's all, I guess we're all about, you ask a question solidly and then you get an answer and then you deal with that answer. So I think so long as it's a clear no, actually a clear no, you hope to have that answer before phase 3, obviously, because that's quite an expensive way to fail.
Yes.
But yeah, certainly you can learn a lot from a clear no that you can then inform the way you're going to approach your next project.
No, absolutely. And your position at the company is as Chief Scientific Officer, Claire, and there are always different sort of shapes that role takes in different companies. So tell us a bit about what you spend most of your time on and how your day looks.
So I don't know if I have a typical day because we're a small company, and so if you ask anybody in a small company,
Of course,
it's quite varied. So it ranges from bringing the milk in the morning through to discussing new projects. So I'm involved in a lot of the early project discussions. So we have a really, really good scientific liaison called Martin, who I work very closely with the initial conversations, kind of introducing our capabilities, and obviously that our capabilities won't be a match for everybody, but very often we find that people are excited by what we can do, and we'll then take them through more details about how we might be able to help them answer questions. So I spend a lot of time helping to develop those proposals and then costing them up. Also, sort of aside from looking for new projects, it's having oversight of our existing work.
yes.
So I have a great team of scientists that I work with who will be managing on a sort of day-to-day level all of the work that we're doing. So I get to kind of check in that everything's working okay, help troubleshoot if things are looking tricky, hopefully also share in the celebrations when things are looking good. Also line management, so trying to make sure that the team are happy and that as a group we have everything that we need. Future planning, where do we go next? We're looking to expand, so we have recruitment ongoing. We're looking to expand in terms of space as well, so I'm looking at that.
Which I understand isn't easy at the moment.
It's not, no. Fingers crossed, it's looking quite positive that we're going to get a little bit more space.
Good.
But yeah, no, it's really varied, and it's kind of having oversight of all of the work that is currently ongoing, with also an eye on where should we be going, where do we need to go in the future. The work that we do, it's really important that we are at the forefront of model development. So we're constantly looking at how do we build on what we can already do. We don't want to sort of stand still. There's still a lot more work that we can do, and we try and learn from what's out there in the literature. We can't do everything though, so it's trying to partly predict what we think is going to be of interest to other people who we're speaking to and other potential partners, but also from our perspective, where do we believe that the next advances are going to come. So it's a kind of mixture of sort of guesswork to an extent, personal interest as well, because there are many different things that we can do, and I think it has to be, life's too short to do a job that you're not enjoying. So we focus a lot on our own sort of interests and where we believe the field is going. So it's good. It's very varied. There's no time to be bored.
No, absolutely. And I suppose, there's a lot of scientists probably listening to this who see this as really the ideal role for them, because you don't move too far away from the science, but you're still exposed to all the strategy and the business side of things and the commercial side of things, as you talked about, but you stay connected to what's happening in the lab. And it sounds like a lot of fun, actually.
Yeah, I mean, I guess that's the advantage of being in a small company. it's the quality of our work and the kind of cohesion of the team is really important. So yeah, it's important to me to be quite engaged with what's going on.
Yes.
That's the exciting bit anyway. That's where I started as well.
No, absolutely. Well, that's a perfect link, actually, Claire, to take us back to talking about your career. So thank you very much. So we do always like to talk about where things started from, and obviously, early on you trained in pharmacology, but going back even before that, for you personally, why science? Why pharmacology? Why drug discovery and development? Why this career for you?
So from when I was at school, I don't think it was necessarily obvious
Okay.
that I would end up doing what I'm doing now. So I was kind of an all-rounder. I was okay at science, but I wasn't strongly sort of only science driven. I quite like writing. I really like languages as well. And I think I like to understand how things worked. So that's why I like languages as well. I like to see patterns between the different languages and where did those languages evolve from. I did do some science at A level as well, and I had the opportunity to do work experience at Glaxo at the time, Glaxo Group Research. So that dates me in Ware, because I grew up in Hertford, which is just kind of next door. So I did a week's work experience when I was in lower sixth, so it's year 12. And that's basically where I had my first experience of what a job in science would actually look like.
Right.
And I think I'd really thought about it before, but it seemed really cool. I got basically given to an industrial placement student for the week, and I kind of shadowed him in his job. So he showed me around the labs, he took me around all the different aspects of the company, introduced me to loads of people, told me about he was doing neuroscience as a degree course. And I thought, oh, that sounds cool, because I like to understand how things work. I'd love to know how the brain works. But that's kind of why I ended up doing neuroscience, because I thought I'd always been brought up to kind of follow your interests, basically. And I thought, that sounds really interesting. I'd love to know how the brain works. So I actually went into doing neuroscience, which was my undergraduate degree, thinking I'd love to understand consciousness. It then took me not very long at all to realize that that's not my strength. So I'm not, I like to understand how things work and fit together, but in quite a reductionist way. And actually, if you want to understand consciousness, it's all about kind of flow diagrams, or certainly at that stage, it was, and I found that very frustrating. And actually, where the things that I found easier and were more naturally kind of fit the way I work was things like pharmacology, and which was thankfully a big part of the course that I did.
Yes.
So that's how I kind of came to pharmacology. I did an industrial placement year as part of my undergraduate, which is something that had been recommended when I was on work experience. So I think work experience is so important. I was 16, 17 when I did that work experience, and I would say that's why I'm where I am now.
Yeah, okay, interesting.
But I was advised to do a placement, because then you can spend a year seeing whether you actually like doing the job. So I spent a year in Paris, which was good, because that, another advantage of science is that there's lots of opportunities to live in different places. So I got to do some languages as well. Spent a year working at Servier in Paris. So not too shabby, spend a year in Paris.
Yeah, it's not bad. Not bad.
Learned a bit of French. Discovered that actually I really liked lab work, which again wasn't something that I necessarily expected, but it was very different to the lab practicals. So I wasn't so keen on lab practicals because you know what the answer is. But when you're doing your own experiments, I like that.
Okay, yeah.
So, and that's why I ended up doing a PhD. So it kind of started from the work experience, got through to PhD in pharmacology, because I'd kind of now cottoned on to the fact that I'm better with reductionist things rather than kind of big, trying to figure out consciousness. So then did PhD in Cambridge in the Glaxo unit.
Right.
So,
yes.
And that was called, so that doesn't exist anymore, which is a real shame, because it was a Glaxo funded unit embedded in the University of Cambridge in the
Mhm.
pharmacology department. So it's called the Glaxo Institute of Applied Pharmacology. I was just a normal PhD student, but my supervisor was Glaxo.
Oh, okay, I see. Yeah.
So it was a normal academic PhD, but with a structure from industry.
Yeah.
And that wasn't so common at the time. And I think
No.
it meant that it was broader. There was much greater focus on why are you doing these experiments? What is the applicability?
Hmm.
And also quite a lot of emphasis, I think, on trying to get broadly trained PhD students. So at the time,
Right.
it was more common to have, you're an electrophysiologist, you just do that. Whereas we all of us in the unit had access to a huge array of different techniques,
Yes,
which was
I see.
a brilliant sort of training ground.
Mm.
And lots of met loads of really good people who have come out of that group. So really enjoyed my time there. It was
Mhm.
quite cellular. So I was doing ion channel pharmacology.
Right. Yeah.
Then, if I carry on kind of through my CV, went on postdoc in Marseille.
Yes.
So I was keen. As I said, I think one of the good things about a scientific career is that you can have the opportunities, particularly early on, to work in lots of different places, which I think is, well, I was really keen on because I wanted to experience different things. But I
Yeah,
think
absolutely.
it does really help to kind of broaden your general awareness of different ways of working. You get the chance to live in a different place. I mean, I got to live in the south of France for 2 years, so that was cool. Met my husband there, so it's definitely worth going. Polished my French a little bit.
Yes.
It was an academic postdoc, but funded by GSK, so it was GSK by that point.
And did you find, from a sort of, I suppose, a scientific culture point of view, you mentioned sort of different ways of working or what have you, were there sort of marked differences? Did you find working in France as opposed to the UK?
Kind of, but actually I think that was maybe more because it was an academic unit.
Okay.
Compared to, so I guess I'd come out of the hybrid industry academic environment, and then I had gone into, it's a CNRS lab. So it was quite different. I don't actually know how much of that was French versus English, so much as just the different kind of type of work.
Yeah.
I guess the biggest challenge is fairly obviously language.
Yeah, of course.
So although I'd spent a year in Paris, I then hadn't spoken French for very long. And I went over to Paris with GCSE French, which was not a massively high level.
No.
So I guess one of the big challenges was just trying to be patient with myself, to give myself a break to learn French. I mean, everyone was lovely to me and put up with my awful attempts at French until, obviously, as long as you keep at it, I got to the point where it was okay and I could communicate with people.
Yeah. I think people appreciate the effort. I always remember a friend of mine living in Switzerland. He was a British guy, and he worked really hard to try and learn Swiss German, which is more, from what I understand, more difficult than German. And he got to the point where he thought, right, I'm going to conduct this meeting in German. And he did it for the first 10 minutes until one of his colleagues leaned over and said, we should maybe carry on this meeting in English. But they really appreciated the effort that he made to do it.
I think I may have had similar things to that, actually.
Yeah, and I mean, you mentioned it there. I always recommend to people, especially if they're early in their career, go and work somewhere else, because it does give you a sort of, I suppose it challenges some of the things that you take for granted, right? Like, there are different ways to live, there are different ways to work, there are different ways that people approach things, and it's helpful to open your mind like that.
Yeah, definitely. I think it's always good to work with different people, and people who kind of come from different backgrounds, have different ways of working. It's something that we actively try and recruit for in Talisman, to make sure that we're not recruiting clones of ourselves.
Yes.
And I think that it sometimes can mean that there are challenges, but for the most part, it's a really beneficial sort of team environment where, we know that we have individuals in the team who are really excellent at just focusing in on the critical question, and then there are maybe others who are great at brainstorming and thinking out of the box. And actually, if you can get all of those people together and harness that, as long as it's in a nice environment,
Yes,
I think
course.
that's the way to crack some of the challenges that we're trying to deal with in terms of scientific complexity.
No, it makes total sense. And so postdoc in Marseille, and then decided that it was time to move fully into industry, and I guess you missed the weather as well. Being
Well,
in south of
absolutely,
France.
exactly. So, like, how could I not come back to Loughborough? Hey?
Yeah.
Yeah, I mean, it was a 2-year postdoc, so
Mm-hmm.
my intention had never been to go into academia.
Okay.
I sort of, once I'd kind of decided that I liked research and understand it, kind of cracking problems or trying to crack problems. It was always very much with a view to doing something that had the chance of being tangibly beneficial. So, ultimately, I would like to get to the end of my career and look back and be able to go, preferably, see that drug on the market. I had something to do with that.
Yes.
Failing that, that you see that drug or that those molecules, I was involved in something which helped inform those decisions. I guess that's kind of the aim of many of us who are working in drug discovery.
Absolutely.
So I was never going to stay in academia. When I finished my postdoc, I kind of looked around at what was available, saw a job advert for AstraZeneca in Loughborough. I actually knew a few people who were already there, so I'd kind of exchanged a few messages with them. They were looking for an ion channel pharmacologist, who preferably somebody who wasn't a pure electrophysiologist, but
Right.
had something a bit broader. So that kind of fitted my background really nicely. I applied, I got the job, so that was all really good, and joined the team in the biology team. So that was basically working on projects from target identification all the way through, or supporting projects all the way through lead optimization.
Yes, I see.
And that's where I learned about how a big drug company works and fits together, and also about the drug discovery process. So that's kind of quite fundamental to how I've spent the rest of my career.
Yes.
I think I was also given some really, looking back, really amazing opportunities from a very early stage. So given that I joined with 2 years postdoc, I was given the opportunity to lead the ion channel work that was going on in respiratory and inflammation. So it was an area that they were the head of department was keen to try and expand. But at that point, it wasn't so common to look at ion channels in that context. It was more in neuroscience. So I should say, having a neuroscience background, I was in respiratory and inflammation, which seems slightly random, but this crops up again.
Right.
And it was because of the ion channel background. So I was basically able to use my ion channel knowledge and apply that to the disease systems that I was asked to apply it to. So I worked on chronic obstructive pulmonary disorder or disease, COPD, and osteoarthritis mainly. So I think I learned, apart from meeting lots of people everywhere I've worked, I've met lots of really amazing people who I've tried to keep in contact with. But I kind of learned and found out the importance of meeting lots of people and kind of understand it. I like to understand how companies fit together and who does what, so that I know who to go to if I wanted something done. And it works both ways, because people would then come to me if they wanted things done.
Yeah.
But I think that's one thing that I quite liked from large companies, because there's pros and cons to large and small companies. And one thing I quite liked in a large company was that as long as you know who to go to, actually there's huge amounts of things that you can get done. You can find answers to things really quickly. So I think I started understanding the importance of developing a bit of a network, particularly kind of on peer network. So this isn't necessarily schmoozing with all of the heads of department.
No.
Although, I mean, there's no harm in making sure they know who you are. But just understanding who are the people who are actually getting things done and who can actually help you. And I think that's something that, you know, I've taken forward as well. Not deliberately, but actually looking back, I think that's something that has been quite useful.
Yes, you don't always have to go right the way up to come back down to get things done, right? You can often go direct to the source if you want.
Yeah, I mean, I have had my wrist slapped for trying to go too direct to the source as well, so it doesn't always work. But
which potentially informs your interest in biotech and smaller companies.
Potentially, yes, exactly. I did say there were pros and cons.
Yes, yeah. So you were with AstraZeneca for 4 years, something like
4
that?
years,
Yeah. And then a bit of a change for you.
Yeah, so it on paper probably looks quite a big change. Actually, I was looking to move down south for largely for personal reasons, so to be closer to my partner. And my old PhD supervisor rang me up out of the blue and asked whether or not I could still remember how to use an automated ePhys device or something like that. I was like, yes, I can. He'd just joined a company called Neurosolutions as CEO, and they were looking for somebody to take over the running of the automated ePhys capability. So it was quite a good fit for my background in the sense that technically I could do that. Also that I like a challenge. I mean, he'd worked with me before, so he kind of knew, you know, what I was likely to be able to do. And it, you know, it seemed like quite a good challenge. It wasn't necessarily something that I'd considered doing previously, but location wise it worked, and I was looking for an excuse to move down south. So I was quite sad to leave AZ, actually. I was enjoying my time there. But from a personal perspective, it made sense. So I moved down to the Cambridge area and was at Neurosolutions for a couple of years, where I would do 3 days a week actually running the experiments. 2 days, I was a bit of a one-woman band. 2 days a week doing business development and kind of all of the paperwork to do with the proposals. So I was running HERG screening, so safety pharmacology. It's kind of different, but it's an ion channel. So I was going out and kind of trying to get new business and working with a BD consultant to try and build the automated ePhys screening side of the business. The rest of the company was in Coventry. So Neurosolutions as a company offers services in high-end electrophysiology. So it's the complex, either in vivo or really complex in vitro biology systems.
Okay, yeah, I see.
And all of those guys were based in Coventry. And I was kind of an outpost in Cambridge, but actually co-located with the CEO, just because that's how it worked. So I think while I was at Neurosolutions, it was really varied in the sense that I was kind of covering BD, a bit of accounts, a bit of the admin, all of the stock checks and everything, and also running the experiments.
How did you find that? Because that was quite different for you.
Yeah, I think the hardest thing actually was being largely on my own.
Right, yeah.
So I much prefer working as part of a team. I was co-located with Zentian, who actually is a biotech company that doesn't exist anymore. So I wasn't completely kind of flying solo. But I think that was the hardest thing. I actually really enjoyed the mix of kind of more customer facing work and in going out and introducing, you know, what could we do, and discussing new proposals. This is obviously quite reminiscent of the role that I now have as well. And I quite like the variety of running the experiments, and also the freedom. You know, I could kind of do what I wanted when, so long as I got everything sorted and working okay. So I enjoyed that. Towards probably about a year in, I outsourced myself to Zentian. So because I had the flexibility to do kind of, as long as I was bringing money into the company. So I would do HERG screening for Neurosolutions 3 days a week, and then 2 days a week I would work on the rigs in Zentian's labs, but kind of as a contractor, basically. And then Neurosolutions decided that they kind of reviewed what their footprint was going to be, and they decided that it was too expensive to keep the Cambridge site, i.e.
Right,
me, going. So they decided to shut down the QPatch and the Cambridge site. Offered me the opportunity to go back to Coventry, but given that the main reason I'd taken the job was to be in Cambridge, that didn't really work for me.
Sure.
But thankfully, on that same day, the guys at Zentient offered me a full-time job.
Okay.
To kind of do full-time what I had contracted myself out to do, but also taking on some more of the automated ePhys kind of coordination and management of how that was working.
Mm-hmm.
So, they really helped me out and supported me. And actually, I think you see that a lot around the small companies around Cambridge, that people will really try and help each other out if somebody does find themselves being made redundant.
I think everyone's been through it, haven't they?
So I very much appreciated them kind of bailing me out effectively so quickly.
Yes.
So I was with them for a short period and then joined MedImmune, which actually again was working back with my old PhD supervisor.
Okay.
That's not the only reason for me joining them.
No. But it's a small world, isn't it? It's
It is a small world. So I then joined MedImmune because they had a job opening that looked really interesting.
Yeah.
So I mean that's basically been the way I have chosen different roles or chosen to apply for different roles, has been based on looking for something that seems interesting, where I think there's enough from my background that would allow me to do a good job in the role, but also a lot that I could learn.
Yeah.
So for that role, it was working in large molecule drug discovery. So I'd not worked in antibody drug discovery before, so that was all new. But the core of the sort of pharmacology background, and they were looking for an ion channel pharmacologist specifically, actually fit really well with my background. And particularly they wanted somebody who could do electrophysiology, but not just electrophysiology.
Yeah, okay.
So I went into MedImmune as a kind of project leader and team leader.
And that must have been a really interesting time to be part of that team as well, because obviously they're still doing very interesting things as part of AstraZeneca, but I suppose thinking about the timing of that, they're probably at the peak of their sort of growth, right? As in growing very rapidly, being very successful with the molecules they were developing. Must have been an exciting time to be around that.
It was, and MedImmune was a brilliant place to work. I just keep saying that I've met loads of really good people. I've been really lucky. I think I've worked with some brilliant people. And it was just a really nice environment with really genuinely very smart people who just wanted to crack on and make a difference, and really collaborative,
right.
Really good interactions between the different groups. So I was split between neuroscience and then also ended up back in respiratory and inflammation again. So it was a really exciting time to be in the company. And I learned loads about both sort of the actual antibody drug discovery, but also this how to move molecules forward and how to manage a team within that kind of organization, how to work more in a matrix organization as well. So I've done that. I had experience of that from AstraZeneca, but from a more junior position. So it was great to have the opportunity to be in more of a leadership role. So I was leading the multiple sclerosis work.
I see.
At MedImmune. And so worked really closely with the guys in Gaithersburg, as well as people in Cambridge, and a mix of neuroscience and immunologists as well. So I think that I've really enjoyed the projects that I've worked on where you have neuroscientists and immunologists, because they're very different ways of working, because like neurons stay still. So the cell bodies stay in the same place, and then they communicate largely from a static position, might quickly move around, whereas the immune cells are constantly whizzing around. And I think even just that difference between the two systems means that immunologists and neuroscientists just think about things in a really different way. And so it's been really nice to think about the world more from an immunologist's point of view.
Yeah, I see.
For them, it's really, I think, important to understand where things are happening. And that's also true in neuroscience. We maybe don't consider it enough.
I thought you were going to say for a second that you noticed that immunologists were always moving around.
Probably, they may be running away, yeah.
And then, so I appreciate we're fast forwarding a little bit here, but tell us about how you found your way to Talisman.
So I was approached by the chairman of Talisman, who's a guy called Mark Traherne, who I'd worked with at Neurosolutions and Zentian, or kind of knew him through Neurosolutions and Zentian. He was and still is the chairman of Talisman, and at that point they were looking for a head of research. And so he'd been tasked with kind of looking through his network to see who might be interested and a potential candidate. So I met him for lunch. He asked me what I'd been up to since he last saw me. I ran through what I'd been doing, kind of a bit of MS, bit of pain biology, and he introduced Talisman as a company to me. Asked whether I'd be interested in learning more, because outlined the role that they were looking to recruit for. So I thought that sounds cool. At MedImmune, so I'd been doing a lot of model development, or my group had a lot of the work that we'd been doing was trying to generate systems, but all rodent systems that would allow us to test the new therapeutics that were being developed.
I see. Okay.
And it was a continual source of frustration that we could only use rodent systems. So there are more options available now, but at that point, although there were protocols out there for generating stem cell derived neurons, they're quite involved, and even now I think it's not really feasible to just embark on it on a whim. You really need to be thinking about building a whole team with a starting point of a couple of people who really know what they're doing.
Yes.
And that's where Talisman comes in. So I thought, well, the idea of having all of our work, like if I joined Talisman, all of the work would be in human, really translationally relevant, patient-derived systems. That would be cool. So, I mean, that's kind of what sucked me in initially. So, and going back to this, looking for a role where there's things that I can bring, but things that I can learn. I joined Talisman without any knowledge of stem cell systems.
Right, yes.
Beyond, having had a bit of a look. I also hadn't done a huge amount of work in Alzheimer's. But felt that I could bring some of the drug discovery experience and approaches. It's how do you approach setting up a model? What questions are we trying to ask? So I met the team, chatted to everybody, thought that the work that Talisman were doing was really exciting. They obviously thought that I could help them because they offered me the job. I accepted. I joined.
There you go. And it sounds like it's been a really interesting ride since then. And so looking back on all of that, Claire, you've probably touched on some of this already, or you've almost certainly touched on some of this already. But if you were to pick out maybe 2 or 3 things that you feel you've learned along the way from a career point of view that I guess you'd share with people, or if you met yourself starting again that you wish you'd known back then, what would have been those career lessons for you, do you think?
I think one of the big ones is that I've always learned the most when things were the most difficult.
Okay. Yeah.
And I try and remind myself of that when you're in the middle of something which feels really difficult. Because looking back, those are the times when it's scientifically and personally, I think I've learned the most. And actually, they're kind of the most satisfying when you do actually resolve the issues.
Yes.
They also try and maintain communication in the good times and when things are going. I'm going to focus lots on things going wrong. Obviously, lots of things do go right.
Of course.
But trying to make sure that you keep communicating with people, because I think often when things go wrong or if there are issues, it's actually because people feel that they're out of the loop and don't know what's going on.
Yeah.
So trying to make sure that everything, good and bad, is clearly communicated, and that kind of goes from all the way from feedback. So we try and give feedback to people as immediately as possible, both good and, not bad feedback, but kind of good and constructive feedback, and keeping people updated. So focus on communication. And I think, sorry, go on.
I was going to say, just picking up on that, I suppose, whether it's investors, or whether it's sort of the leadership team, or whether it's collaborators or partners or whatever, I imagine you find as well that as soon as you start to share the challenges, then they start trying to help, right?
Exactly. I mean, we're all in it together. There's not a kind of them and us. So it's just trying to be open. That doesn't mean communicating everything.
No.
It's not like, oh no, I dropped a pipette on the floor, sort of thing.
Yeah.
Although, I mean, we do need people to tell us if the pipette's been dropped on the floor. But yeah, it's kind of just making sure that there's enough communication so that people, say our collaborators, understand what's going on. If there are ever delays, it's communicating that, what is going, when should they expect to hear from us?
Right. Yes.
What has happened. It's not that we've just gone radio silence and are ghosting people. That's not really on. That doesn't help. And if I met myself, going to your question, I think the big one I would say is just try not to overthink things and try not to worry too much, because you can only do your best. So if you go into the situation trying to do your best, then if you make mistakes, it's not the end of the world. Try not to make a mistake more than once.
Sure.
But people will make mistakes, and it's just to try and do your best, but don't beat yourself up about it if things go wrong. Because I think I and many others sometimes dwell on the things that haven't worked when actually it's not necessary. Things go wrong, life goes on.
Yeah, and like you said, you have to have things go wrong to learn from them. I think people tend not to learn as much when things are going well, because typically you sort of can get away with cutting a few corners if the majority of it's going all right. So it's when things fall apart that you start to realize, okay, well, maybe this would have been done better this way, or maybe I should have looked at this. Or
absolutely.
So, yeah, it's I think you often see that the most successful people are the ones who've had the most things go wrong, but they've just kept going.
Yeah. No, I mean, you've just got to stick at it.
Yeah, absolutely. And you mentioned, coming back to Talisman, you mentioned that it's an exciting time, that there's lots of exciting stuff on the agenda. What's next? What does 2023 look like for you?
So we've got some really exciting projects lined up, which I obviously can't tell you about, but we're really keen to get started on them. We're recruiting, so we have a project leader position and cell culture scientist position that's live at the moment, and we'll likely be recruiting again next year. So we're trying to gradually expand the team. We're looking to take on a little bit more space. We're generating new models, doing a bit more BD. So it's kind of more of the same, only better.
Yes. Okay. Well, it sounds really interesting. Best of luck with it.
Thank you.
I'm assuming that if people fit those descriptions, then they should look at where to find you.
Indeed, on our website.
Yes.
Would be great.
Thank you very much, Claire.
Thank you, Tom.
Thanks for joining us on Careers in Discovery, and don't forget to subscribe for more insight into the world of drug discovery and R&D. Do take a look at our sponsors, Singular Talent, and their mission to make hiring better for companies and individuals in drug discovery and R&D. You can find them at www.singulartalent.io. See you next time.